I like to talk. That's probably not much of a surprise to you considering you're watching me talk right now, but I generally think it's one of the most unique things about being human. And it's pretty easy to argue that speaking is what led to the transfer of knowledge between generations, which has led to every other technological advancement, including whatever you're watching me on right now. But for a long time, it wasn't clear exactly what made us able to speak. That all changed in 2001 when a specific family called KE in the literature was discovered. In this paper, 3 generations of the family was studied, and you can see here that the filled symbols in black are the ones who were affected. And with genetic analysis, they found it was a single gene that was responsible for this condition, called FOXP2. Members of the family with this condition had 2 different types of problems. There's physical problems, such as with fine orofacial movements, but they also had problems with their grammatical skills. And the study also found an unrelated patient who they called CS, who had a very similar condition and the same genetic change. And this was a fantastic boon for science, because one of the big problems with studying genetics is that most traits have many, many genes involved. So finding one gene that had such a big effect gave researchers in the future a really good place to start. Now, on a physical level, we've known since 1991, thanks to this paper, FOXP2 and FOXP1 cooperatively regulate lung and esophagus development. Well, we've known that FOXP2 helps with esophageal muscle development. Title kind of gives this one away. But after 2001, the research into FOXP2 kicked into a much higher gear. This paper from the following year was looking into whether humans had multiple different versions of the FOXP2 gene, and it found that there's no amino acid polymorphism. Now, this is pretty significant because when there's no polymorphism, it means that one gene is very strongly selected. By contrast, something like eye color has polymorphism because there isn't that much genetic advantage between having blue or brown or green eyes. And what's even more fascinating is the paper indicates that this is a relatively recent development in our evolution. This comparison table shows the amino acids of FOXP2 of 6 different species. Humans and mice diverged from each other about 70 million years ago, but there's only 3 changes in our FOXP2 gene. This change here, and these 2 on the 4th line. However, 2 of those changes are divergences between us and chimps, and we are much more closely related, only diverging a few million years ago. We don't know exactly when these changes happened, but the evidence from the paper shows that it was strongly selected for something like 200,000 years ago, which is maybe not coincidentally around the time of anatomically modern humans. However, there is some evidence that Neanderthals also had this mutation, so the jury's still out on that one. But whenever it happened, it soon became clear that FOXP2 wasn't just important for how we speak. In 2004, this team published this paper about FOXP2 expression in birds. And a crocodile. Okay, before we go on, I just want to say science is so awesome that this group of people was like, we want to study birds, and let's throw in a crocodile for good measure. Anyway, in the birds it was discovered that FOXP2 was expressed more in the days when vocal learning occurred, leading them to believe that FOXP2 is associated once again with vocal plasticity. Another team published a paper the next year in 2005 showing that mice vocalize differently if they have a damaged FOXP2 gene. Disruption of both copies of the FOXP2 gene caused severe motor impairment, premature death, and an absence of vocalization. They also discovered that disruption of even one of the 2 copies of the gene causes problems. In order to test directly the genetic effect of these 2 changes unique to humans, mice were genetically engineered with both of them, and they were found to have increased neural plasticity. Their brains were completely different from wild-type mice. But at first, it wasn't clear which of the 2 genes had the big effect, or maybe it was both. But in this 2023 paper, 2 mice lines were independently created, the T303N substitution and one with the N325S substitution. The mice with the N324S substitution were remarkably similar to the wild type, but the mice with the T302N substitution were dramatically different from the wild type, meaning that of the 2 mutations, T302N is responsible for the enhanced synaptic plasticity. To explain how crazy this is, I need to explain what T302N means. There are 21 alpha amino acids, and those are the ones that create the proteins in your body. T302N means that a threonine at position 302 changed to an asparagine. And to show you what a small change that is, I actually downloaded the genomic data from the National Institutes of Health for both humans and chimpanzees. Here is the protein encoding for FOXP2 for chimpanzees on the left and humans on the right. Now this looks really long and scary, and to a certain extent it is, but this shows all the different isoforms, so it's all the different ways that this gene can be read. Let's go ahead and simplify it. Now here we have pan-troglodytes, the chimpanzee, on top, and Homo sapiens, humans, on bottom. You'll notice they look very similar. And now I'm going to show you what this change that was so momentous that led to all of human speech and technology and history and civilization, I'm going to show you exactly where it is. That's it right there. This T turning into this N. This one letter into that. That's insane to me. That's the tiniest difference that I can imagine, almost. But I do mean almost. Because if you look at threonine and asparagine, the difference between the two of them is just here. Threonine has 1 hydrogen and 1 oxygen. Asparagine has 2 hydrogens, a nitrogen, and an oxygen. Which means the difference between these two amino acids, the change that has made all the difference, is just 2 atoms, 1 hydrogen and 1 nitrogen. And if that's not the coolest thing that you've heard all day, post anything cooler in the comments, I dare you. Anyway, so that's why we can speak. Thank you and your 2 additional atoms for watching. Bonus fun fact for staying to the end of the video. Threonine is normally encoded by 4 different codons, which is a combination of 3 different nucleotides, including ACA, so adenine, cytosine, adenine. Now asparagine is only coded by 2 different codons, but one of them is AAC. So it's very likely that this mutation happened just because one nucleotide switched with the one next to it. So like,